All right, active transport processes. So, active transport is all about the movement of molecules against the concentration gradient by using energy from ATP. We often say that with this, we go, you go against the concentration gradient. You can say you go up the gradient. You can also say you go from a low concentration to a high concentration. When we were doing passive transport, you go from high to low. If somebody sprayed some perfume all in this area would be very, very concentrated with perfume. And ultimately, it would start to diffuse throughout the entire room until it reached a state of equilibrium where all parts were equal. That is passive transport, no energy required. Not to get a little gross, but if you were in an elevator and you let one rip, you wait for those few seconds until it's diffused through the elevator, then everybody starts sniffing and looking at you. It's kind of nasty. That's why if you're in an elevator with one other person, you might not want to do it because obviously it was you. But that's an example of diffusion. This works with light. It works with molecules. It works in either liquid or gas. It's just the movement of molecules to reach an equilibrium. This is the opposite of that. This is active transport. And one of the most popular examples of this is the sodium, which is Na, potassium, pump. And because you see those positive charges, that means that these are ions. The sodium ion, potassium ion, pump. So as you just saw in the video, in a nutshell, show a little phospholipid bilayer here. This is the surface of a neuron. I want you guys to know what a sodium potassium pump actually does, and then we're going to talk about what its role is in all of active potential in the body. Someone in first period goes, I get it, but like, where is it used? What is its purpose? I mean, that's a good question. Well, what does it do? We're going to talk about what it does. So it does not look like this in nature. This is just meant for study purposes. This is called a protein pump, or another word you could use is a carrier protein. Now, as you all can see, it goes through the membrane from one side to the other. So would this be known as a peripheral protein or a transmembrane protein? This is a transmembrane protein. And whenever you see the word pump or carrier, those two words are screaming at you active transport. Whenever you see pump or wherever you see carrier, that means that it needs energy in order to be used. It requires ATP to operate. So generally, guys, as you saw in the video, every little lime green circle I'm going to draw here is going to represent a sodium. There is some sodium on the inside of the cell, which is the bottom, but there's a lot more outside. And then every little triangle I'm drawing is going to be potassium, and I'll write a key up here somewhere. There's a lot of potassium on the inside. Not a whole lot on the outside. So a brown triangle is going to represent potassium. And a green circle is going to represent sodium. All right. You will have 3 sodium ions at a time go into this carrier protein, this sodium-potassium pump. And next, I'm going to redraw it again. So try to do this fast. It's going to change its orientation because it now has a phosphate. That little purple dot there is a phosphate, and I will elaborate on that phosphate momentarily, but just know that that phosphate belonged to ATP. And ATP is energy. This is what the mitochondria produces. It's why the mitochondria is called the powerhouse of the cell. You've been hearing that since middle school. It makes ATP, which is cellular energy. And when it is being used by the pump uses the ATP, these 3 sodiums are going to exit the cell and go out into the extracellular space. While at the same time, 2 potassiums are going to come into the pump and try to get to the inside of the cell. So this is all just I'm throwing a lot at you. We're going to slow things down after I get this out. Now, that phosphate breaks off when the sodium-potassium pump is turned off, OFF. It's opening towards the inside of the cell. There is no phosphate that is turning it on yet. When the pump is turned on, it is opening up towards the outside of the cell. So right now, it's in the off position. It is releasing those 2 potassium ions to the inside of the cell. And the whole process repeats itself. So I'll let you guys get all caught up here. When it's on, it's facing the outside of the cell, like here. You see that purple dot? That is a phosphate from ATP. Notice that I do not have that purple dot on it on these 2 images on the left, because on the 2 images on the left, it's facing into the cell, which is down. That's when it is turned off. The ATP is what turns it on. Yeah.
Does it return to naturally off position?
Yes. When there's no phosphate, it automatically goes back. So it's like this, guys. Look at me. Off, on, off, on. And when the phosphate detaches from it, that's what makes it turn off again. So speaking of phosphate, speaking of ATP, let's quickly go over what that means. What is ATP? ATP is adenosine triphosphate. You see an A, you see a T, you see a P. ATP. Now, the way that ATP actually looks, it is an adenine molecule, like the same ones that you have from DNA and RNA. Adenine, guanine, cytosine, thymine, uracil. You have an adenine attached to a ribose sugar. Ribose is the sugar in RNA. It's a pentose. And then you have 1, 2, 3 phosphates. This is what ATP looks like, energy of the cell. A lot of videos that you watched, they just really simplify to look like this. That's it. Now, I want to really focus on this phosphate, the third one. That third and final phosphate is called the terminal phosphate. This is where all the power comes from. Whatever cellular process gets that phosphate, it is going to be energized. It's going to be turned on. And what do you suppose ATP is called when it loses that third phosphate and it's down to 2 phosphates? Do you remember from Honors Bio? ADP, adenosine diphosphate. This third phosphate detaches and it's going to attach to some pump somewhere. And that process is called phosphorylation. If this ball of yarn is a phosphate from ATP, and Dan is a pump. You ready? You ready? You ready, Dan? You ready? You have been phosphorylated. And then it's going, I want you to just go crazy. Wave your arms. Ah, then it goes off. Off. On. Ah, off. That's how it works. This phosphate will reattach to ADP to make it become ATP again. So it is recycled. This goes back to the first law of conservation of mass. Mass is not created or destroyed. It just gets recycled. Same with energy. Okay. Dan, thanks for spazzing out there for the betterment of your classmates. Now, the pump actually has a name. The pump is called a sodium, potassium, ATP ACE. When you see the suffix ASE at the end, what does that mean? It's an enzyme. Most enzymes are named after what they do. And based on what you saw, how many sodiums per how many potassiums are exchanged? What's the ratio? 3 sodiums, 2 potassiums. That's the ratio. For every 3 sodiums that are released from the cell, 2 potassiums are brought into the cell. Okay, so I'm going to try to do this fast, guys, but not too fast because we are out of here at 1:03 today, and it's already about 12:42, so it's roughly 20 minutes. That's not a lot of time. So this is actually a 6-part process. I'm going to try to do this quick. Let's do part 1. Here we go. Three sodium ions enter the pump. Just by the way I drew it, is this pump in the on position or the off position? That's right. Now I'm going to try to stick with the colors. Green is going to be sodium. Brown is going to be potassium. Okay, that's just part 1. Part 2. A phosphate from ATP phosphorylates the pump and activates it. Okay, notice that there's now a phosphate attached to it. That phosphate came from ATP. Maybe I can condense this into 5 steps. We'll try to do steps 3 and 4 together. Step 3. The 3 sodium ions go against the gradient and exit the cell. The cell in this case is a neuron. I'll get to that. They exit the cell where there are more sodium ions. So you're going from a low concentration to a high concentration. Low to high. In order to go against the gradient, you have to have energy. 2 potassium ions enter the pump. Okay? So I'm pretty much blending steps 3 and 4. All right, so the 3 sodiums are leaving. They're on their way out. And the 2 potassiums are coming in. Step 4. The phosphate detaches from the pump. We call this dephosphorylation. Whenever you put the letters DE in front of a word, you're going to do the opposite of what the root word is. The pump returns to its off position. And I forgot to put a phosphate on part 3, so if you want to color that in real quick, there it is. Okay, the phosphate is off. And what do you think is going to happen in the 5th and final step? What's going to happen to those 2 potassiums? They're going to move into the cell. All right. That's a lot, isn't it? Let's you all get caught up. Does anybody need me to zoom in on 3 and 4? Just 5? Yeah. Number 2. The sodium potassium pump is turned on. It is facing the outside of the cell. and it's about to release the 3 sodium ions that it has. Yeah, Jet. The sodium potassium pump is turned off, and it is carrying 2 potassium ions, which it is about to release to the inside of the cell. Okay. Number 5, the 2 potassium ions have left the pump, and they're going to the cytoplasm of the cell. Okay, yeah.
So does the phosphate stay with the pump?
No, the phosphate will go back to an ADP to make it become ATP again.
But it stays with the pump.
It stays with the pump while the pump is turned on.
Okay.
Yeah.
What function does ADP have that does not?
Say that again?
What function does ADP have that does not?
ADP is like a dead battery. It really doesn't do anything. It just serves as a conduit to have the 3rd phosphate reattached to it. When it's ATP, it's energized. It's powered. When it's ADP, it's a dead battery. It doesn't do anything. It's just waiting to get its phosphate back. All right, so now what we're going to talk about, and we have about 10 minutes left, we're going to do an action potential, guys. Try to get this in within 10 minutes. So when you do an action potential, it's depicted in a graph, X and Y axis. The x-axis is time, and that time is going to be measured in milliseconds. This happens very, very fast. A milli is 1/1000th of a unit. And on the y-axis, we have millivoltage. We're going to start at the bottom with negative 70 millivolts, then negative 50, around negative 50, then 0, then positive 40. Now, when a nerve impulse, an electrochemical signal travels through your nervous system, it goes nearly 250 miles an hour. This is how you can have such a quick reaction time. And when you're playing ping pong and maybe a sport, video games, they really try to sharpen your senses because you have to be very observant of your surroundings. You know, Andre Vasilevsky, the goalie of the Lightning, the way he practices his reaction times and his really conditions his nervous system to react quickly. So he plays a lot of ping pong because you got to move so fast in a very short amount of time. Well, an action potential looks like this. We're going to go through all these different parts. The blue part here is going to be a resting potential. I'll do that first. Can you turn off the light? The orange part is going to be depolarization. The green part is going to be the action potential. The pink part is going to be repolarization. And the yellow part is going to be the sodium potassium pump. So we have a lot to get through, not a lot of time to do it. So here we go. I'm going to try to keep this color coordinated. Number 1. This is all about sending a signal through your entire nervous system, either from your brain or to your brain. Resting potential. Here's what you need to know. It is at negative 70 millivolts. That's the inside of the neuron. A neuron is a nerve of the nervous system. You need to get it all the way up to positive 40. Right now it's negative 70, so it is resting. The inside of the neuron, when I say inside, I mean in the neuron, is mostly potassium, some sodium. Outside, mostly sodium, some potassium. Okay? That's pretty much it. You're resting. You're relaxed. Your nervous system is not stimulated. But when I went, ah! Earlier today, some of you jumped because that was a stimulus. And your whole body reacted to it. That was your muscles moving your body. That was prompted by an action potential. Now, number 2, you have depolarization. In depolarization, sodium ion channels open. Sodium ions move into the cell. The voltage increases. It's becoming more positive. So guys, right now, there was a lot of sodium outside the neuron. The channels to allow sodium in are open. So all that sodium is going to start to rush into the neuron itself. Now, something else I think it would be a shame to neglect is as more sodium enters, more sodium channels open. So you can let in even more and even more and even more and even more. Any questions so far? So right now you're just letting sodium in. That's it. Sodium is just rushing in. Step 3 is an action potential. This is very short-lived. Imagine feeling how you felt when I screamed, your whole body was rattled. Imagine feeling that way all the time. That would be sensory overload. So this is very temporary. This has a millivoltage of positive 40 millivolts. You have the maximum amount of sodium and potassium inside. So this is very short-lived, like maybe a millisecond at most. This is what is allowing the electrochemical signal to travel down your neuron to whatever the intended target is, your brain, your spinal cord, your muscles. Okay, number 4. This is called repolarization. Okay, now pay attention. And remember, you guys need to study this stuff. You can't just get it by sitting here in this class. You need to work it out. So here, potassium channels open. Potassium exits the cell. The polarity or the charge becomes more negative. It decreases. So you're going back down. But here's what I want you to, this is where you get by studying. You're going to realize you started with sodium outside, potassium inside. Now, as I speak, right now the potassium is on the outside, the sodium is on the inside. So it's flipped from how it was when we started. So that gets us to our third and final step, the sodium potassium pump. 3 sodiums at a time are returned outside the cell. And how many potassiums are brought in? 2 potassium ions at a time are returned inside the cell. Now, the sodium-potassium pump is what type of transport system? Active or passive? So what does it require in order to operate? Whenever you see active, you need to think of energy. So right now, the neuron has returned to a resting potential. And what was the millivoltages of a resting potential? Negative 1, or excuse me, yeah, negative 70. I was going to say 170. So it's like, imagine in order for this classroom to be stable, all the girls were outside, all the boys were inside. This room is going to get activated. So all the girls are outside, all the boys are inside. All the girls come inside too. So now the room is all boys and girls. We're maxed out. And then we're going to depolarize. All the boys go out and all the girls stay in. The reverse of what it started as. So now you need to get all the girls back out and all the boys back in to get things the way they were. The pump helps put things the way they were in order to get back to the resting potential. Because you don't want your nervous system to be stimulated 24/7 when it's being stimulated by some sort of sensory input. Hearing, tasting, smelling, touching, seeing, any one of those. Yeah. So there's the graph. And as you can see, following the sodium-potassium pump, you're back to resting potential, and it just recycles itself. Okay.